Good afternoon, everyone. My name is Leslie Meltzer, and I'm the Chief Medical Officer at Orchard Therapeutics, and I'm really excited to share with you what we are doing today in the cell and gene therapy space. Here are our forward-looking statements, which I'm sure you have all read. So Orchard is a company that came into existence built on a foundation of hematopoietic stem cell gene therapy with the goal to end the devastation caused by genetic and other severe diseases through the curative potential of this approach. And shown on this slide is an example of a family that has been impacted by our therapies, a family with metachromatic leukodystrophy. The oldest sibling is on the left. She was a normally developing child who experienced symptoms and then a very rapid decline at the age of 5. Subsequent to this photo that was taken about a year ago, she has since unfortunately died. Her younger brother, who was also diagnosed with this disease, was able to receive gene therapy before the onset of the disease, and he is now doing very well, completely absent of symptoms, and in junior high today in the United Kingdom. And this is just an example of the type of impact our therapies can bring to patients. So we leverage the hematopoietic stem cell platform as our approach, and we're capitalizing on two important characteristics of hematopoietic stem cells. First is the fact that they are self-renewing. If you provide these cells to a patient, they will form a durable repository of stem cells, presumably for the lifetime of the patient. Secondly, they are able to give rise to all of the cells of the hematopoietic lineage, including erythrocytes, cells that produce platelets, and cells of the immune system, including, importantly, white blood cells that are able to infiltrate and reside in the tissues where you have the opportunity to directly correct disorders that impact those tissues through overexpression of enzyme and cross-correction. So we work off of an autologous hematopoietic stem cell platform where we harvest the patient's own blood stem cells, genetically modify them with an integrating lentiviral vector, re-administer these cells following a conditioning procedure where they will then engraft in the marrow and give rise to genetically corrected blood cells of all of these different lineages and allow us to correct disorders that impact the brain, the gut, and other organ systems. This is our pipeline today. Our late-stage pipeline is predominantly focused on neurometabolic diseases. Our most advanced program, which I'll talk more about in a moment and already spoke a little bit about, is LibMeldy for the treatment of MLD, which is currently approved in Europe, and we are actively seeking to file our BLA at the end of this year or beginning of next year. Our next most advanced programs are a program in Hurler syndrome, MPS I, which is entering registrational trials next year, and then a program in Sanfilippo syndrome, MPS IIIA, that will be reading out proof of concept data later this year. We also have a number of early stage preclinical programs that are leveraging this platform for the treatment of disorders in the central nervous system, including progranulin deficient FTD, as well as immunological disorders such as genetic subtype of Crohn's disease and hereditary angioedema, and I'll talk a little bit more about that in a bit. When we look at our company kind of short-term and long-term plans, in the near term, our focus is really executing on those rare diseases and those rare neurometabolic diseases, MLD and the MPS indications, and build out those capabilities on the regulatory, manufacturing, and commercialization and access side on this platform. Over the longer time frame, we look to, again, expand this approach into larger indications, use this technology to enable other modalities of treatment, which I'll talk a little bit more about in a bit. And these are areas where we are actively seeking partnership opportunities for indications where there's a compelling clinical and scientific rationale. So as I mentioned, LibMeldy is our lead program. It is approved in Europe for the treatment of early onset metachromatic leukodystrophy. This is based on a study that was conducted starting in 2010, so the longest follow-up now is in patients that were treated over 12 years ago. And what you can see from this clinical data is that we're able to preserve both motor function, which is shown on the left, and normal cognitive development, which is shown on the right. And again, we have children now where we followed them for over 10 years, where they were treated as infants and toddlers and are now middle school or high school students, some of whom have shown no manifestations of the disease since the time they were treated. So based on these data, we have a regulatory approval again for the treatment of early onset MLD. And our focus right now in terms of launching in Europe is really on these three key pillars. The first is securing market access and reimbursement in each of the key geographies. The second is on identifying patients and facilitating the adoption of newborn screening. So I alluded to this in the beginning, that patients have the best outcome when they're treated very early in the disease course, preferably prior to the onset of symptoms. And today, the way you find these patients is because they have an older sibling that was diagnosed with the disease, and then they can be tested to see if they are also either carriers or have two deficient copies of the gene and therefore will develop the disease. The goal would be that we can move toward identifying all patients at birth through newborn screening and therefore have a much larger population that would be eligible for therapy relative to today. And then, of course, we want to be able to enable treatment of patients that are identified in any market where we have an approved product and build out the infrastructure that allows these patients to be treated. So where we're at today, we're actively securing reimbursement throughout Europe. We have formally secured reimbursement in 3 countries, the UK, Italy, and Germany. The list price in Germany is over 2.4 million euros. The prices in the UK and Italy have not been publicly disclosed, but they are in the same domain, and we also have a reimbursed early access program in France. Since the start of the year, in the first half of the year, 3 patients were treated in the commercial setting, and we expect to treat even more patients in the second half of the year. We've also established 5 treatment centers throughout Europe in the 4 countries I already mentioned, plus the Netherlands, and we've initiated newborn screening pilots throughout these countries as well. Our most advanced pilot is in Germany, where over 50,000 babies have been screened, and I'm very pleased to say that we have word that there is a potential positive baby that was identified in that screening pilot, which is hopefully going to allow us to have a data package that would support implementation of national or US state level screening down the road. Finally, we're focused on expanding our footprint both from a market access and a QTC standpoint, and also, again, gearing up to potentially launch in the United States following the successful submission of a BLA. Our next most advanced program, again, is in Hurler syndrome, MPS IH. This is an indication very similar to MLD, a little bit more slowly progressing, and also more systemic manifestations of the disease, including skeletal abnormalities, corneal clouding, and a number of other disease features that are burdensome to the patients. There is a current standard of care with hematopoietic stem cell transplant, which is associated with suboptimal outcomes, both from an efficacy standpoint, these patients continue to decline, as well as from a safety standpoint, with mortality in the range of 5 to 20% and a number of adverse events that can have a chronic impact on the patient's quality of life. So we've initiated this program with a proof of concept study that read out earlier this year and was published in the New England Journal of Medicine, where 8 patients were treated with gene therapy. They continue to develop on a normal course with improvements in cognition, growth, skeletal health, joint mobility, and a number of other features. We are in the process of designing a registrational study that will allow us to demonstrate superiority versus the standard of care, and the goal would be to initiate that study next year. I wanted to share, there we go, some images that I think really articulate or demonstrate the potential power of this platform. So on the left-hand side, you can see 2 patients that were treated with gene therapy prior to treatment. And then on the right-hand side, you can see what these children look like a year or a year and a half later. And you can see marked improvement in the physical features of the disease, including joint contracture, spinal curvature, kyphosis. Even the facial features of this disease, which are very characteristic, have improved over the year of follow-up following a single administration of this therapy. And this is the kind of image that we put up, and we hear feedback that those physicians who treat with the standard of care today never see results like this with the current standard of care. So a lot of excitement about this. I'm really eager to bring it into the registrational international multi-center study next year. When talking about our pipeline, again, trying to move or expand beyond the rare neurometabolic space and capitalize on this platform in a number of different indications where we believe the biology of the disease is amenable to this approach. And our priorities at the moment are, again, this genetic subtype of Crohn's disease, progranulin deficient FTD, and a program in hereditary angioedema, which is being conducted in partnership with Farming. We're also looking to broaden this application and leverage genetically modified HSCs for other approaches besides simply providing a working copy of a deficient gene. So for example, we are actively exploring the use of this platform to provide engineered Tregs for autoimmune diseases such as multiple sclerosis, as well as genetically modifying these cells to deliver a vectorized antibody that would allow us to provide antibodies to difficult to access tissues that these HSC-derived cells can infiltrate, such as the CNS, to allow potential for oncological indications in the brain. The ability to expand beyond the ultra-rare disease into these larger indications does require a few advancements that we are also actively working on, including expanding our platform to allow for large-scale manufacturing, including things like stable cell lines for the manufacture of viral vectors and closed systems for the transduction of the genetically modified cells and the use of transduction enhancers to reduce the amount of vector required to produce cells for each patient. We are also looking at opportunities to employ reduced toxicity or brain-specific conditioning regimens that will allow patients to receive and engraft with the genetically modified cells, potentially with a better safety profile than what we have today. So like I said, we're actively seeking partnerships to expand the work in these different areas in the preclinical setting. We already have the partnership for our HAE indication, and we have the opportunity to partner these ongoing programs as well, and the work in the technological advancements. So as we look ahead over the next few years, our main goal for the first half of this year was establishing our commercial footprint and securing reimbursement for Livmeldy in Europe, which we have indeed completed. Before the end of the year, we are planning for a pre-BLA meeting for Livmeldy in the US, with the goal of a submission either end of this year or beginning of next year, reporting out our data in Sanfilippo syndrome, and reporting out preclinical data in NOD2 Crohn's disease. We are also planning in 2023 to initiate our registrational study in MPS IH. And then finally in 2024, we're targeting for submission of an IND in NOD2 Crohn's and advancements of our preclinical pipeline. Very exciting time, very excited to see how we are able to leverage this platform toward a variety of different indications and modalities and really transform the way some highly unmet needs are addressed. And with that, I thank you for your time.